The future of neuromuscular disease treatment is unfolding before our eyes. At the 2025 MDA Clinical and Scientific Conference, researchers revealed groundbreaking advances in gene therapy, exon skipping, anti-inflammatory treatments, and precision medicine. Let's break down the biggest scientific discoveries from this year's event. Starting with Duchenne muscular dystrophy, gene therapy and exon skipping trials continue to push the boundaries of treatment. Let's dive into the major updates. First up is RGX-202, an investigational gene therapy by REGEN-XBio. Findings show increased dystrophin production in DMD patients. Interim results indicate functional improvements noted early, but long-term safety is still under assessment. However, challenges remain, including the risk of immune response and the need for further dose optimization. Next, we have D-Landystrogyne moxaparvovec from the EMBARK study by Sarepta Therapeutics. Findings reveal sustained functional benefits and muscle MRI improvements. Long-term data over 2 to 3 years show treated patients maintained ambulation longer than untreated groups. Safety-wise, a mild to moderate immune response was observed. This data confirms that gene therapy for Duchenne muscular dystrophy is making progress, but researchers continue to fine-tune safety and efficacy. Exon skipping in Duchenne muscular dystrophy is showing promising results. In the DYNE-251 study by Dyne Therapeutics, a phase 1-2 trial in boys with exon 51 mutations has demonstrated significant increases in dystrophin production at higher doses. Safety profiles show no serious concerns, only mild side effects. Similarly, the WVEN-531 study by Wave Life Sciences, known as the Forward 53 study, reports significant dystrophin production increase at 24 weeks in treated patients. Phase 3 trials are the next steps. Exon skipping is proving to be a key approach for patients who may not yet qualify for gene therapy. Now, let's delve into the recent breakthroughs in spinal muscular atrophy therapy. The Rainbow Fish trial, focusing on risdiplam in pre-symptomatic SMA infants conducted by Roche, has shown promising results. After 2 years, treated infants maintained their motor milestones. This reinforces risdiplam as an effective long-term therapy for SMA. Next, we have apitegromab from the Sapphire trial, targeting SMA type 2 and type 3. Findings indicate increased motor function and reduced fatigue in patients. With these positive outcomes, an FDA submission is expected soon. The long-term effectiveness of SMA treatments like risdiplam and apitegromab is shaping the future of disease management. Turning our attention to amyotrophic lateral sclerosis, or ALS, let's explore some innovative treatments. Radicava ORS, developed by Mitsubishi Tanabe Pharma, has shown that after 96 weeks, patients experienced a 30% slower decline in ALSFRS-R scores. This helps extend the quality of life for ALS patients. Additionally, tofersen, a therapy developed by Biogen and Ionis for SOD1 ALS, has demonstrated prolonged survival and stabilized respiratory function. Notably, it is the first targeted therapy for SOD1 ALS patients. ALS treatment is advancing with targeted gene therapies and biomarker-driven research. Gene therapy for LGMD is making strides. Pfizer's LGMD2E gene therapy has shown sustained dystrophin expression for up to 24 months. Biomarker improvements have correlated with muscle function preservation, indicating significant clinical impact. Myonexus's LGMD2D gene therapy findings are also promising, with 70% of patients showing increased sarcoglycan protein. Gene therapy for LGMD is progressing, with long-term follow-up studies showing sustained benefits. Let's delve into the latest advances in therapy for chronic inflammatory demyelinating polyneuropathy, or CIDP, and myasthenia gravis, also known as MG. The ADHERE trial, focusing on FcRn inhibitors for CIDP, has shown significant promise. Findings indicate a substantial reduction in relapses among CIDP patients. The next steps? Well, FDA approval is anticipated. For myasthenia gravis, FcRn inhibitors have also demonstrated encouraging results. Patients experience sustained symptom improvement and a decreased dependency on steroids. These therapies are indeed reshaping the treatment landscape for CIDP and MG. Looking ahead, cutting-edge therapies like base editing, prime editing, and CRISPR are paving the way for permanent gene correction in DMD, SMA, and ALS. Cytosine-base editing for DMD has shown precise exon editing in preclinical models. First-in-human trials are expected soon. Neurofilament biomarkers for ALS and MG accurately track disease progression and hold potential as early intervention tools. The future is bright for neuromuscular research, with new innovations bringing us closer to precision medicine solutions. That wraps up our deep dive into the biggest breakthroughs from MDA 2025. If you found this update helpful, don't forget to like, subscribe, and hit the bell icon for more cutting-edge research updates. Let us know in the comments, which breakthrough excites you the most.